Hello everyone. In this video I'll be covering the JNK signaling pathway as it relates to axon degeneration, and I'll finish the lecture with by reviewing this fantastic paper published in the Journal of Neuroscience by Larhammer et al. Titled The ST20 Family Kinases MAP4K, MEEK1, and TNIC Converge to Regulate Stress-Induced JNK Signaling in Neurons. So I hope you enjoy. So JNK stands for C-Jun N-terminal kinase, and it's a highly conserved and critical stress signaling kinase family, and it's involved in neurodevelopment, axon regeneration and degeneration, and neuronal degeneration following injury or disease. So you can tell by its name, C-Jun N-terminal kinase, that it's a kinase that was originally discovered for its role in phosphorylating C-Jun, which is a critically important transcription factor. There are 3 isoforms of this JNK protein, 1 through 3, and they collectively have about 10 different splicing isoforms that can generally, it really complicates things. Most neuroscientists only really worry about JNK2 and JNK3, which appear to have low basal activity, are primarily activated by cellular stress, and most importantly, they are the primary isoforms expressed in the brain, especially JNK3. Knockout studies of JNK3 suggest the protein is important for apoptosis induction during development, but it can also exacerbate neurodegenerative phenotypes, so its function is broadly related to promoting apoptosis, whether it's during brain development or if it's a pathological condition during neurodegeneration. JNK inhibition has been shown to reduce amyloid beta 42 plaque formation and plaque load in Alzheimer's disease. And I should mention Alzheimer's disease models in mice. And it's also been shown to reduce mutant Huntington accumulation in mouse Huntington's disease mouse models. Complete knockout of JNK causes a severe neurodevelopmental phenotype due to improper cell migration and improper axon guidance. So it's an important protein. So what then activates JNK? Well, this signaling pathway seems to respond to many different stressors, including growth factor deprivation, DNA damage, ER misfolding, and mitochondrial dysfunction. So pretty much whenever there's cellular dysfunction in any different way, we usually have JNK signaling accompanying it. And in terms of where this happens inside of the cell, well, JNK3 is localized to mitochondria, where it facilitates the phosphorylation and often the activation of apoptotic stimuli. And it's also found translocated to the nucleus, where it promotes C-Jun phosphorylation and the formation of this complex called AP1, which is kind of the activated transcription factor of C-Jun. So let's take a closer look at this JNK pathway. Okay, so let's start with some of the cytoplasmic activity of JNK. So we're looking at JNK, and we're looking at some phosphorylation targets. One really important phosphorylation target is a protein that is found on microtubules. So this will be our microtubule, and attached to our microtubule is going to be dynein. Dynein is our retrograde motor protein that's found on the microtubule. An important protein that is associated with dynein, called BIM, B-I-M. BIM is associated with dynein, and what happens is when JNK is active, it can phosphorylate dynein, and it actually the phosphorylation of dynein dislodges BIM from dynein. So BIM gets detached from dynein when JNK phosphorylates. And BIM is a BH3 protein that will then go to the mitochondria, and it inhibits a class of protein called BCL2 type proteins. So BIM inhibits BCL2 proteins. Let's draw this over here. So this is our mitochondrial membrane, and we have these BCL2 proteins that are embedded into our mitochondrial membrane, and we'll get into exactly what BCL2 proteins do in a second, but for now, we're going to stop here and say BIM inhibits BCL2 proteins. We have to look at another BCL2 protein, or BH3 protein that is unleashed by JNK. And this happens on another phosphorylation event happens on the motor protein of actin, which you might remember is myosin. Myosin is the motor protein attached to actin. And attached to myosin is a protein very similar to BIM. It's BMF. BMF is commonly attached to myosin. In a similar mechanism, we can see JNK phosphorylating myosin when it's active, and the phosphorylation of myosin detaches BMF. BMF is another BH3 protein that can go and inhibit BCL2. So we have BIM and BMF being basically freed by JNK phosphorylation so they can go and inhibit BCL2. So why do we care if BCL2 is inhibited? Well, BCL2 inhibits a very important apoptotic process, the oligomerization of Bax and Bac. Bax/Bac oligomerization. So Bax and Bac are two proteins that are expressed in the mitochondrial membrane, and they like to oligomerize, but they normally cannot oligomerize because BCL2 is constitutively inhibiting them. When BCL2 is instead inhibited by a BH3 protein like BIM or BMF, it allows these Bax and Bac proteins to oligomerize in the mitochondrial membrane, and when they do this, they form a pore. They make an opening in the mitochondrial membrane, and this opening in the mitochondrial membrane releases cytochrome C. And I won't go into the specifics of apoptosis, but this is how apoptosis starts, is that Bac or Bax oligomerizes, forms a pore, and this allows the contents of the mitochondrial matrix to exit the mitochondria, and one of the most important ones is cytochrome C. It induces the formation of the apoptosome, cleavage of caspase 9, then the effector caspases 3 and 7. See, JNK also phosphorylates. Let's see. I'm going to have to erase some of this. So, another important protein that JNK phosphorylates to help induce apoptosis is a mitochondrial bound protein that's very similar to BCL2 called MCL1. MCL1 is a homolog of BCL2 and performs a very similar task. MCL2 inhibits Bax and Bac, right? That's what MCL1 does. It's just like BCL2. It's of the BCL2 class. However, when it's phosphorylated by JNK, a little p, it gets phosphorylated. This phosphorylation of MCL1 recruits a protein called MULE. MULE stands for MCL Ubiquitin Ligase. I don't know what the E stands for, but it's an MCL ubiquitin ligase, so it targets MCL1 for degradation. Right? When MCL1 is phosphorylated, it produces a binding site for MULE, which then ubiquitinates MCL1, leading to its degradation in the proteasome. And by effectively deleting MCL1, it's promoting the oligomerization of Bax and Bac and the release of cytochrome C and apoptosis. So these are some of the more direct mechanisms by which JNK promotes apoptosis. Okay, so in this slide, we're going to go through a couple of additional mechanisms by which JNK indirectly promotes apoptosis. Doesn't directly activate a BH3 protein, but perhaps might favor the apoptotic choices of a cell. So one of the important phosphorylation targets, as we alluded to earlier, is the in the nucleus. So say this is the nucleus, and JNK can enter the nucleus where it phosphorylates C-Jun, right? C-Jun, because JNK stands for C-Jun N-terminal kinase. And when C-Jun is phosphorylated, it can bind this other protein called ATF2. So the phosphorylation of C-Jun allows it to bind ATF2, and this forms the functional AP1 complex. It's called AP1. And it binds DNA, and I won't get into the specifics, but it influences gene expression. And, this would be highly cell type specific and depend on chromatin formation and many other factors, co-transcription factors. But generally, the AP1 transcription factor is how JNK influences gene expression that, could be apoptotic. It might not be. It depends. Another important phosphorylation target of JNK is this protein called SCG10. SCG10 is a microtubule stabilizing protein. So SCG10 stabilizes microtubules, and when it's phosphorylated by JNK, it leads to the dissociation of SCG10. And so through this mechanism, JNK promotes the depolymerization of microtubules. Depolymerization. So again, JNK phosphorylates SCG10, causing it to dissociate from the microtubules, and this promotes the depolymerization of microtubules, which is an important process in something like axon degeneration that we're going to talk about in a second. JNK also indirectly promotes apoptosis by inhibiting a protein called NMN-AT2. This is a protein intricately tied to many different metabolic processes because it produces NAD through the NAD salvage pathway. And I won't get into the specifics of that pathway. I do have a lecture on it. But NAD, briefly, is a molecule that can be reduced into NADH. And NADH is the electron donor for complex 1 in the electron transport chain. So NAD is basically required for the electron transport chain to work. And so if JNK is inhibiting NMN-AT2, then it's going to be causing NAD, I'll say NAD depletion. So JNK leads to NAD depletion by inhibiting NMN-AT2. The depletion of NAD leads to energetic failure of the cell. It leads to cell death and the activation of this other protein called SARM1, which I will discuss later in this lecture, but it leads basically to these different effects. So the mechanism isn't really understood exactly how JNK is inhibiting NMN-AT2. I don't know if it's ever been looked at, but it's probably through some kind of phosphorylation event. But just to summarize, we get metabolic failure because NAD depletion. We see microtubule depolymerization because JNK is phosphorylating and removing SCG10 from our microtubules. And we see gene expression through the AP1 heterodimer of C-Jun and ATF2. Okay, so in this slide, we're going to cover some of the mechanisms by which JNK is activated. So this is going to be our synapse, or synaptic bouton. And when we're talking about JNK activation, the upstream activator of JNK is DLK. And DLK is going to be the focus of the current paper that we're going to review next. But briefly stated, DLK activates JNK. And when JNK is activated by DLK, which is via phosphorylation, I should mention. DLK is a kinase. It's part of the MAPK family of kinases. It phosphorylates JNK. JNK can then associate with another protein called GIP3. GIP3 stands for JNK interacting protein. And so GIP3 is a JNK interacting protein 3 that bridges JNK to dynein. To dynein. So dynein is our retrograde transporter that's going to move this new complex. So JNK GIP3 dynein is then going to move back towards the cell nucleus. That's where we get all of our different phenomenon. We get SCG10 inhibition. We get SARM1 activation, which accelerates the loss of NAD. We get CJUN activation. We get BMF and BIM activation. So we see all these different effects. We get MCL inhibition. We see cell death. And again, DLK phosphorylates JNK. This allows JNK to bind to SCG10 so it can get moved back towards the cell nucleus where it can actually start, doing these different activities. One of the other ways in which JNK is activated, or I should say one of the ways in which JNK is held in check, is constitutively inhibited, is through this other protein called AKT. AKT, also protein kinase B. Everyone just calls it AKT. And AKT is a kinase that inhibits JNK. It constitutively inhibits JNK. However, it also inhibits DLK too. So if AKT is on, it inhibits JNK and DLK and prevents the JNK signaling pathway. However, AKT is only active under favorable conditions. Specifically, it's only activated, in the synapse, it's primarily activated by TRKA. TRKA is a growth factor receptor. And specifically, it's a growth factor receptor that binds to nerve growth factor. Nerve growth factor is our primary neuronal growth factor that will bind to TRKA. It causes TRKA to dimerize and phosphorylate each other, and it basically leads to the activation of AKT. So when there are growth factors, NGF when it's present, or BDNF through a TRKB, they indirectly lead to the inhibition of DLK and JNK through the activation of AKT. So the big question, though, is what is upstream of DLK? What activates DLK? And that's going to be the big question that's tackled in this current paper is what exactly is upstream of DLK. So through this negative regulation, AKT inhibits them, but what leads to DLK activation? Okay, so now that we have a decent understanding of the JNK pathway and how it relates to axon degeneration, we can move into reviewing this other paper. This paper is titled The ST20 Family Kinases, MAP4K4, MINC1, and TNIC Converge to Regulate Stress-Induced JNK Signaling in Neurons by Larhammer et al. So as we discussed earlier, JNK is the upstream activator of JNK, at least when we're talking about axon degeneration in response to growth factor deprivation. So JNK activation is actually sufficient to induce JNK signaling. And so understanding how DLK is regulated could lead to possible therapeutic targets, and that's what the current paper is aimed at identifying. So in order to do this in an unbiased manner, they first took a model of DLK and JNK activation, which was simply trophic factor withdrawal. So they're depriving neurons of trophic factors, and then they are screening. They screened for thousands of enzyme inhibitors for their ability to downregulate C-Jun phosphorylation. So C-Jun phosphorylation is a downstream effect of DLK, JNK activation. And they used all these different enzyme inhibitors to see if any of them were capable of preventing C-Jun phosphorylation. And they landed on 3 kinases, MAP4K4, MINC1, and TNIC, as they blocked C-Jun phosphorylation, and they even prevented axon degeneration following nerve growth factor deprivation. And when they probed the kinases a little more closely, they discovered they were upstream of DLK, and they were responsible for activating DLK. Although the specific mechanism was not investigated as to how these different proteins were activating DLK. So let's go into the specific pathway. Okay, so we start with our synaptic bouton again, and what we are really examining is the activation of DLK. So the critical player here is going to be DLK activation. And typically what is believed to activate DLK is kind of the negative regulation of DLK by AKT. Typically, we talked about how nerve growth factor can activate TRKA, and the activation of TRKA leads to the phosphorylation and activation of AKT, which inhibits DLK. So by the conventional idea is that the loss of nerve growth factor, which is the model they're using, indirectly leads to the activation of DLK, because you lose AKT signaling, you lose TRKA. So if you remove nerve growth factor, the loss of this repression leads to DLK activation. That was the idea, but there might be a more active mechanism of activating DLK. So instead of simply losing the inhibitor of DLK, maybe there's something that actually directly activates DLK. And indeed, they found that these 3 proteins, these STI-20 family of proteins, are responsible for, are in some way upstream of DLK activation, and their kinase activity, when inhibited, downregulates DLK activation. We have MINC1, and we have TNIC. So these 3 proteins converge to activate DLK. And again, the specific mechanism isn't investigated. They aren't known to directly phosphorylate DLK. It could be directly phosphorylating. It could also be recruiting DLK and promoting DLK dimerization, which is required for DLK activation. So it could be that they maybe autophosphorylate themselves, and maybe they induce DLK recruitment and dimerization. And it could be there could be some intermediate proteins in here. They don't necessarily know. But these 3 proteins are upstream, and they mediate DLK activation. And just a reminder, DLK activation is what's responsible for phosphorylating JNK and allowing JNK to associate with the dynein gyp3 JNK retrograde transport complex that allows this complex to move back retrograded towards the nucleus. So without DLK, JNK isn't associating with dynein and gyp3, and it's not being transported back towards the nucleus to mediate all these pro-apoptotic effects. So One other thing that these authors found is that none of these three proteins are alone responsible. They can, if one of these is knocked out, the other two can compensate. So really, to get complete DLK inactivation, they needed to knock out all three of them, not just one or two. All three of them kind of function redundantly to activate DLK. Okay, so on this slide, we're going to go through some important figures for the paper. And I wanted to start with this Western blot. So after doing their high throughput screening for enzyme inhibitors that reduce phosphorylated C-Jun, they landed on these three inhibitors that inhibit the class of MAP4K4. So there's a protein called MAP4K4, but also it's the name of the class. And when they were looking specifically with these inhibitors using a more sensitive Western blot, they found that phosphorylated C-Jun is downregulated when each of these enzyme inhibitors were supplied. And you can see this is the control condition right here. This is how much C-Jun we should expect under normal conditions, but that amount is vastly downregulated when these enzyme inhibitors are supplied. So in panel B, what we're looking at here is they're looking upstream of C-Jun. They want to see, is it C-Jun that's losing its phosphorylation, or does that problem, can we relate it upstream to JNK? And so they're looking at these same enzyme inhibitors. I'm not exactly sure why they used GNE 495 instead of these two, but nevertheless, they're looking at the amount of phosphorylated JNK. And this is what we should expect. These are two different isoforms of JNK, I suspect. And that's the normal amount of phosphorylated JNK. When the MAP4K4 inhibitor is supplied in this lane, we see vastly downregulated phosphorylated JNK. In fact, it's about the same amount of phosphorylated JNK that we see during baseline. So on the far left, right here, this is how much baseline phosphorylated JNK there is just under normal conditions. And when this inhibitor is supplied, we see it return down to baseline. So it's basically, it pretty much completely removes the phosphorylation from JNK. And this condition over here, this is a phosphatase that is known to target JNK, and they use this as a control condition to make sure their antibody towards phosphorylated JNK was specific for phosphorylated JNK. Now on the right, we see these findings in graphical format. Phosphorylated JNK intensity, you can see in these two conditions with the MAP4K4 inhibitors, we see it returning to baseline. It's about as much phosphorylated JNK as in the control condition with nerve growth factor. And we see a similar trend with JNK in the cell bodies down here. When the enzyme inhibitors of these MAP4K4s are they're preventing C-Jun phosphorylation, they're preventing JNK, and you can even go upstream to DLK. So the reduction in the JNK activation goes all the way up to the most upstream regulator, the DLK protein. So these inhibitors are inhibiting something that is upstream of DLK. In this slide, we see the use of floxed MAP4K4, and we see that when CRE is administered, we will have deleted our MAPK4, and when nerve growth factor is removed from these cells, we see axon degeneration. And in this next condition right here, we see axon degeneration when TNIC is suppressed and when MAP4K4 is deleted with CRE. Right? And in this condition, we're looking at something similar. We see MINC1 being silenced And we see, again, MAP4K4 being floxed and deleted with the addition of Cre. However, we see something interesting in this condition. We do not see axon degeneration. And what's happening in order to accomplish that is that they have knocked down TNIC and MINC1 with siRNA, and they have removed MAPK4 by floxing it and adding Cre. So what this slide is saying is that in order to completely suppress DLK activation and thus allow neurons or allow axons to survive, they needed to knock down TNIC, SMINC, and MAPK4. So in order to get a complete suppression of DLK and the JNK signaling pathway, in order for axons to survive, they had to knock down all three of these upstream kinases. So in this last figure that I've included, we see that MAP4K4 inhibitors prevent DLK and phosphorylated JNK translocation to the nucleus, which is kind of the critical event in this whole process. See under normal conditions that when nerve growth factor is removed, right? These three are when they're removed. Normally, when nerve growth factor is removed from the media, you get lots of phosphorylated JNK in the nucleus and lots of DLK that has moved to the nucleus. However, when you inhibit your MAP4K4s using, it's kind of hard to read here, but this is a MAP4K4 inhibitor. This is another MAP4K4 inhibitor. You do not see the translocation of phosphorylated JNK into the nucleus. Maybe a little bit, but not nearly as much as compared to those conditions. And you also don't really see any activated DLK in the nucleus either. And same thing with these inhibitors. So really the idea here is that we're seeing less activated DLK, we're seeing less activated phosphorylated JNK, and we see less of it in the nucleus. So that explains why we see less CJUN phosphorylation. And ultimately, these inhibitors are targeting the MINC1, the TNIC, and the MAP4K4. So that is indicating that these are the upstream activators of DLK. And so they don't necessarily identify how these MAPK4 or these STE20 family of kinases are activating DLK, but they do identify them and implicate them as being upstream of DLK. So I hope you guys enjoyed this presentation. If you have any questions about the paper, I'd love to answer them, and thanks for watching.